Please explain in detail the classical genetics, such as law of segregation, law of independent assortment, statistical calculations, genetic mapping, patterns of inheritance, etc. Classical genetics, also known as Mendelian genetics, refers to the study of heredity and the underlying principles governing the transmission of traits from one generation to the next. This field of genetics was pioneered by Gregor Mendel, an Austrian monk, who conducted experiments with pea plants in the mid-19th century. His work laid the foundation for understanding the basic principles of inheritance, including the law of segregation, law of independent assortment, and more. 1. Law of segregation. Mendel's first law, the law of segregation, states that during the formation of gametes, reproductive cells, each individual inherits two alleles, versions, of a gene, one from each parent. When gametes are produced, the two alleles for each gene separate and end up in different gametes. Thus, an offspring inherits one allele from each parent, randomly determining the expression of a particular trait. 2. Law of independent assortment. Mendel's second law, the law of independent assortment, explains that the inheritance of one trait is independent of the inheritance of another trait. This means that the alleles for different genes are sorted into gametes independently of each other. However, this law is applicable only when the genes are located on separate chromosomes or are far apart on the same chromosome. 3. Statistical calculations. Statistical calculations are essential in classical genetics to predict the frequency of specific traits in offspring. The Punnett square is a widely used tool for illustrating the possible combinations of alleles from parents, helping to determine the probability of a particular phenotype or genotype. The chi-square test is another statistical method for evaluating the consistency of observed data with expected Mendelian ratios. 4. Genetic mapping. Genetic mapping is the process of determining the relative positions of genes on a chromosome. This is achieved by analyzing the frequency of recombination, or crossing over, between different genetic markers during meiosis. A genetic map is measured in centimorgans, cm, which is a unit of recombination frequency. Genetic mapping is crucial in identifying the location of genes responsible for specific traits or diseases. 5. Patterns of inheritance. Classical genetics describes several patterns of inheritance based on the interaction of alleles. A. Autosomal dominant. A single dominant allele from either parent is sufficient for the expression of the trait. Autosomal dominant traits typically show a 3:1 ratio in a monohybrid cross, A. B. Autosomal recessive. Both parents must contribute a recessive allele for the trait to be expressed. Autosomal recessive traits show a 1:2:1 ratio in a monohybrid cross, A, and a 9:3, 3:1 ratio in a dihybrid cross, AABB. C. Codominance. Both alleles are expressed simultaneously, resulting in a distinct phenotype for the heterozygous individual. For example, in human blood types, the IA and IB alleles are codominant, resulting in the AB blood type. D. Incomplete dominance. In this pattern, the heterozygous individual exhibits a phenotype intermediate between the two homozygous phenotypes. For example, in snapdragons, a cross between true breeding red, RR, and true breeding white, RR, flowers produces pink, RR, offspring. E. Sex-linked inheritance. Traits determined by genes on the sex chromosomes, X and Y, show sex-linked inheritance. X-linked recessive traits are more common in males because they have only one X chromosome, while females have two and can mask the effect of a recessive allele. In conclusion, classical genetics provides a foundation for understanding the inheritance of traits and the principles governing the transmission of genetic information from one generation to the next. With its fundamental laws and patterns of inheritance, this field has significantly contributed to our knowledge of genetics, paving the way for more advanced techniques and applications. 1. Multiple alleles. In some cases, genes can have more than two alleles, leading to a greater diversity of phenotypes. One example is the ABO blood group system in humans, which has three alleles, IA, IB, and I. 2. Epistasis. Epistasis occurs when one gene modifies the expression of another gene. This interaction can lead to complex inheritance patterns, such as coat color in mice, where the expression of one gene can mask the effect of another gene. 3. Polygenic inheritance. Some traits, like human height or skin color, are controlled by multiple genes, each contributing to the overall phenotype. These traits typically exhibit a continuous distribution in a population, as they are influenced by multiple genetic and environmental factors. 4. Environmental influence. The environment can play a role in determining the expression of a trait. For example, the color of hydrangea flowers is determined by both genetics and the acidity of the soil in which the plant is grown. 5. Pedigree analysis. Pedigree analysis is the study of the inheritance patterns of a trait within a family. Pedigrees are visual representations of the relationships between family members and can help trace the inheritance of a particular trait or disease through generations. This tool is essential in understanding the mode of inheritance and identifying carriers of specific genetic disorders. Classical genetics has provided a robust foundation for our understanding of heredity and the mechanisms governing the transmission of traits. This knowledge has not only contributed to advances in genetics and molecular biology but has also had a profound impact on medicine, agriculture, and other fields. The principles of classical genetics continue to inform modern genetic research, helping us to understand the complexities of gene interactions, inheritance patterns, and the role of the environment in shaping phenotypic outcomes.